Hello, this is BrainCan channel, the channel about brain research. Welcome. My name is Vasilisa Zhukova, and today we have a special episode. Today we will not talk about the brain, we will talk about DNA. So, DNA was the most influential discovery of the 20th century. It changed our perception about the organization of life and information storage. With time, damage and mutations occur in DNA. Some of them don't have a significant impact. Some of them are lethal. Especially this is important if we want to understand and treat such pathologies as cancer, schizophrenia, and dementia. To be honest, genetics was always beyond my grasp. But fortunately, our today guest is Anastasya Stolyarenko. She's a Russian molecular biologist who studies DNA damage, repair, and replication of damaged DNA. Thank you, Nastya, for coming. I'm pretty sure that today you will help us to look at DNA from a new angle.
Thank you for having me, Vasilisa.
So the first question will be very naive. What is DNA damage?
The DNA, it is present usually in the center of any cell, most cells, especially maybe for erythrocytes in the blood. But anyway, it's present in most of the cells of the body or of unicellular organisms like bacteria. Even though it's in the middle, it's surrounded by the cytoplasm, it still gets attacked by various DNA damaging agents. For example, it's UV light or X-ray. Everybody knows, everybody is nervous when they go to X-ray studies for their lungs or something, that they can have a certain level of DNA damage after this examination. So these are like maybe natural DNA damaging agents, but also we have a lot that are connected with cars, fuel exhaustion, fuel that's burnt in cars, or like smoking. All of this produces DNA damaging agents that can attack the DNA. And so DNA damage is when something happens with the constituents of DNA, which are nucleotides, or the structure of DNA, which is a double helix. So when something happens to the helix. So your question was what DNA damage is. Yeah, so probably.
So can you please extend this a bit? For example, is this psychological stressor a factor of DNA damage? Because currently it is known that stress, especially psychological, affects everything. So maybe it's true also for DNA damage, or it's not known.
I'm sure that it is known, but I'm not quite sure if I know anything about this connection. Stress elicits adrenaline, and I can imagine that it can cause certain changes eventually to DNA via some DNA pathways, but I'm not I'm not familiar with any.
Okay, thank you. So then what happens next? I think if the cell have damage in its DNA, what's the fate of this cell? Can it continue its living, or it is a big problem for the cell? So what's the coping strategy of the cell?
Yes, the coping strategy is usually DNA damage response, but a lot of factors can influence this. First of all, it is the type of DNA damage, because yes, first of all, it is the type of DNA damage and the timing of DNA damage. So this timing is it concerns the time during the cell cycle. So you know that the cells they divide, most of the cells, but a lot of the cells in the brain they don't.
Yes.
Yeah. They have their and cells depending on whether they are dividing on or in which phase of the cells cycle they are, and they have different coping strategies with DNA damage, and also different types of DNA damage have different type of DNA damage response. For example, DNA damage caused by UV light, like TT dimers, it's the damage that happens to so DNA is a long molecule, usually megabases, so millions of what is called bases or nucleotides, but there are just 4 types of them. So just 4 types of letters that we have is A, G, T, and C. These are the bases in DNA, and these are what mostly gets damaged when we talk about DNA damage. But different DNA damaging agents, they attack certain DNA bases. So, for example, UV light frequently causes a link between 2 standing T's, 2 adjacent T's. And so this is such a frequent type of damage, and animals are usually exposed to UV light, that there exists an enzyme that treats these cross-linked these bases. I'm not sure that this can be termed a crosslink, because no, this is not a crosslink though. They're just adjacently linked, or maybe yes. Maybe. This is an intra-strand crosslink. PT dimers are intra-strand crosslinks in one strand. And so there is an enzyme, so a protein in the cell that can recognize and unlink these 2 T's. So now this damage can be replicated safely. If it is not replicated, then there happens replication arrest happens. And the cell can solve this problem in various ways. So cells that are not dividing, like dormant, they can be dormant. They can stay in this state for quite a long time, and they can accumulate DNA damage without much bad effects for the cell. But when the cells divide, they can't tolerate having most types of DNA damage because the big problem with DNA damage is that it can't be replicated. So if you're a cell and you need to divide, you need to first replicate your DNA. And DNA
Wow.
is
So that the cell dies or it stops the replication?
yes, it stops the replication and it has time to do something with this damage so that it can be replicated, you see. And
Mm-hmm.
so first of all it tries to uh, like call the enzyme that can do something with this damage directly, but not for many damage types. There are these direct enzymes that can recognize this DNA damage and treat it. So usually there are more universal systems, like systems that can excise this base from the DNA.
Mm-hmm.
That is called a base excision repair, and this is a second type of DNA repair. So the first one is direct repair, and then base excision repair. So there
So
also
that means that this enzyme just comes, recognize that, I don't know, imagine we have this helix, and if we have this link, it means that we don't have now the ideal helix. So they start to have some conformational changes, right? And the enzyme recognizes it, that this is not an ideal helix anymore, and just it cuts out this damage, right?
this is one type of
Mm-hmm.
repair is when the helix is distorted, but
Mm-hmm.
sometimes it is not distorted
Wow.
when the damage, because the helix, it has a minor and major grooves. And so when you have DNA damage in the major groove, and it's a small adduct that is
Mm-hmm.
added to a DNA base, then the helix is not distorted. Like
Mm-hmm. Okay.
most alkylation and oxidative damage, it will not distort the helix, but it has its own systems to deal with this type of damage. And so it's one is base excision repair, and the next is nucleotide excision repair, when the whole nucleotide and several surrounding nucleotides are cut out, including the damaged nucleotide. And then this short deletion is
Mm-hmm.
replicated using the other DNA strand. So the DNA is double helix with two strands. And so this really helps to repair it. Another type of DNA repair.
Can you please a bit slow down here? Because you've just mentioned that the replication switches from one strand to another one, right? To another string, to another sequence, which is like a healthy and normal sequence, right?
It's the same sequence. It's the same sequence, but
But
it wasn't. It's
uh,
complementary. It's
mhm.
complementary.
Yes, but it is not. But it includes all nucleotides, and it
Yes.
has a normal, not damaged structure, right?
Yeah, exactly. And so it can be replicated.
So this is very fascinating. Actually, it always hard for me to understand that. Imagine we have only four bases, and just their combination determines our species, our life, the pathologies that we will have, the immune system, and so on and so on. I'm not a big fan of Sapolsky's theory that everything is predetermined, but still DNA and our genes have the major impact, I think, on what we are. What do you think about it?
Yes, of course, you're right. But I do believe that a big part of our existence is not predetermined, because
Yeah,
in genetics, and I think that not many people do realize this, that in genetics there is a very important concept.
Mhm.
I'm not sure what it's in English though. It's called the reaction norm.
What is it? Yeah.
And so the best illustration of this concept that I've ever seen is um, it's a picture of a guy which is divided into halves, and he has different surroundings in two of these parts of this picture. So one, he has good food, a tennis racket, good
Mhm.
sleep, the sun, open windows, and the other, he has cigarettes, coffee, I don't know, alcohol, TV in the other
Mhm.
part. And so this guy, the two halves of him, they look different because
Mhm.
of the surroundings. And so this is a very important concept that we need to know, that we can influence most of the how most of the genes work, because genes are very susceptible to this reaction norm thing. So the eventual phenotype that a person will have depends both on the genetics and the surroundings, and usually this proportion is I don't know, the in many phenotypes it's 50% or even more of what the surroundings can have an impact on. Yeah,
Yeah.
so
So DNA is not a recipe book. It's just an option, a manual. But
no,
then,
it's a recipe book though, but the proportions of the molecules that will be made according to this recipe book, we can determine it by our behavior, by our surroundings. Yeah,
by the way we live, yeah,
yeah,
that's.
exactly.
I think we need to explain a bit that DNA is not always open for the transcription processes, so it's usually in a very tight conformation, and the gene it has to be opened for this big molecular system proteins that will read the sequence. And actually this is why some genes are more susceptible, right, to be open or to be influenced by environmental factors than others.
Yeah, uh, so the reason why DNA is initially in a compact form is that we need to fit in like 2 meters in single little cells, and so that is why it is very compact. That's like of what I imagine is the reason why it had to be compact. But then mm, uh, so it looks like it looks like thread for knitting, maybe like a
Yeah.
ball of thread actually, but it is and so it's maybe many balls of thread that are also compacted into threads of higher and higher dimension. And
Mhm.
so if you look at the smallest thread, you will see that it in the center of this ball of DNA, there are proteins called histones. So they're like the center of the ball, and they were they evolved to be able to regulate the way DNA works because they have tails protruding out of the ball that can have different modifications and call different protein regulators. And sometimes these tails can say, transcribe me. And then the sequence is transcribed better and the gene works better and gives more products. Like an example will be like for the direct repair system I told you. So there is a common DNA damage that can be caused, like for example, it's alkylation. It's alkylation, so it's a CH3 as an alkyl radical addition to G.
The tail of the histone.
No. It's a chemical group attached to G, one of the 4 DNA bases.
Okay.
I'm like going to connect the transcription and DNA damage.
Great.
There's my dog. So when we have DNA damage with alkylation on the G base of DNA, in the 6th position of G, there is an OCH3 group. Like this damage can be caused by temozolomide that is used in chemotherapy for glioblastoma. And so,
brain cancer. Yeah.
Yes, exactly. And so this damage is cured by MGG, a protein that comes and takes this damage off. Like
so it cuts the alkyl group, right?
Yes, exactly. It cuts it off and like everything is normal. And the protein goes to recycling and the damage no longer exists. Everything is okay.
So it sounds fantastic. But I have a very boring question about, I don't know, classifications. So we've just mentioned a set of repair mechanisms, but how do you call it? How do you classify this repair system? Is it a part of the immune system, or you have a separate classification of this? Because I would say, but as I mentioned before, I was always weak in genetics. But I would consider a repair system a part of the immune system that deals with damage and mutation at the level of DNA sequence. But how?
I think immunologists won't agree with us. This DNA repair is a part of molecular biology. Like if you take a molecular biology course, you will have all the processes that happen with DNA, which is DNA replication, DNA repair, and DNA recombination. These are the three R's. And then another thing that happens with DNA, but it's a second part of the molecular biology course, is transcription, because that is the first how RNA is made. So it's the first part of the RNA part of molecular biology. So it fits perfectly. I think that some people can call it an immune system for DNA, but I think it's more of a normal person look at it. Not not a molecular biologist and an immunologist would know where it fits, you see. But from the point of view of a person outside person, maybe you could say that it's an immune system of DNA, for DNA, but like it would be a more uh, maybe you need quotation marks for this.
Generalization of the concept. Yes, I got it. So since you are working at the molecular level, this is your kingdom, and everything that happens at your molecular level is a part of molecular biology.
No, I mean that immunologists, I think they would also not believe that DNA repair is their stuff, because they, in my view of immunology and the person who has had immunology courses, they deal more with these cells that nobody else studies, B lymphocytes, T lymphocytes, and all that. Yeah, you see. So
yeah. Okay, very interesting. Thank you. So I have some concerns about how the repair mechanism is conserved across different species. Is it a very old mechanism, or it evolved together with appearance, with emergence some species?
Well, as far as I know, most repair mechanisms, they should be conserved in eukaryotes, because bacteria, they have some other mechanisms that eukaryotes don't have. Bacteria, they frequently have like source repair that eukaryotes don't, because bacteria, they can have a lot of DNA damage at the same time, and they need to survive at any cost. So they can try to relieve this damage by replicating it wrong. You see, so get a lot of mutations, and that is the reason why bacteria can sometimes survive under antibiotics, for example, that should not let them survive. But some bacteria, because of this source repair mechanism, they can get very, very new phenotypes and can try to survive even under antibiotics. And that is the reason for multi-resistance development that everybody's talking about, and the reason why we need something new, new ways to cope with bacteria now, like phage therapy.
That's incredible. So the logic is that sometimes in the cells you can find DNA damage, and then cell trying to survive, it generates mutations actually.
Yes, right? Of course. Yeah, of course. So the difference between DNA damage and DNA mutations is actually mutations happen when DNA damage has been replicated. So, for example, you had a G in one strand, and it was alkylated, for example, and it failed to be repaired, or it failed to be repaired. It stayed, and the cell failed to replicate it in a correct way, and it replicated not with a C, which is according to the which would be the nucleotide in accordance with the general complementarity rule. Yeah, so we need maybe to explain that further. And maybe because of this, it decided to put some other T there, which the cell would put during replication just to survive, not because it's the correct nucleotide that needed to be there, but this is how it decoded this damaged nucleotide and it put a T. And so the next time when this strand with the T is replicated, then opposite it, there will be A in the first strand, you see? And so this is how mutation is fixed. A mutation is fixed. First we had damage, then it was replicated in the wrong way. And then it was fixed because the next time it was replicated, this was replicated correctly, but the nucleotide was wrong in opposite the damage. And so replication is a key process for DNA propagation, and for our cells to survive. And the reason why we can heal and why we have our skin is constantly healing, because new cells are forming. And so during this process, replication happens all the time. And it happens according to the rule of complementarity. So there are only four letters in DNA, but when one strand is copied, we always have a G, a G standing opposite C, and an A standing opposite T. And so further, there is a C opposite G and T opposite A. Yeah. It is because of their chemical nature, how hydrogen bonds are formed. So DNA is the two DNA strands are held together thanks to hydrogen bonds. Where Vasilisa, this is not for you.
I'm totally happy because I'm a chemist and finally I understand something.
Yeah. So it is for the people who are watching us. Okay, so a mutation is fixed when a damaged DNA nucleotide is replicated in the wrong way. When that should be C, there is A or T or G. You see.
During aging, did you observe on your models what happens with the repair system? Does aging affect somehow the repair system? I think it should, but I don't know.
Well, I know this just from not as a specialist because I haven't encountered systems like this, but I know that in aging, repair works. Repair systems are weaker in aging cells, and so they can cope with DNA damage less. They're less successful in coping with DNA damage, but when repair systems are weaker, there is a second group of systems, which is called DNA damage tolerance. It is a second maybe group of systems, what should I say, when repair didn't work. And that is what I study. DNA damage tolerance.
Rev1, right?
Rev1 is the key protein that comes when repair was not
present
did not occur on maybe in aging cells or maybe in cancer cells when repair systems don't work. They frequently don't work in cancer cells because they're mutated. Cancer cells accumulate so many mutations that repair genes can be affected and not work as effectively. And so Rev1 comes and invites several polymerases that don't care about DNA damage. They can replicate any damaged nucleotide, maybe with a very big adduct, or maybe DNA strands that are, I don't know, something happened to them with very big adducts, like of several phenol rings.
What is the adduct? You mentioned this.
Yes, so adduct is is in addition to a normal DNA base, actually.
Okay.
So,
Okay.
so like a CH3 in
So
alkylation.
alkylation is an example of adapt, right?
Yeah, a CH3 group on G in the position G of 6 of the G base of DNA is an adduct. But adducts can be like, I don't know, several phenol rings connected together, which are like very huge constructs, and normal polymerases that are like, they're very accurate. They only know A, T, G, and C and nothing else. They've never seen that in their life. And when they encounter this terrible stuff, then it's replication arrest. Yeah, it's replication arrest and death actually. So first repair is called, but when repair does not work, like in old or cancer cells, then DNA damage tolerance is called with Rev1 protein. And Rev1 is like a hub. It can try to replicate this damage by itself, or it can call other DNA polymerases that can replicate it because they have very wide active centers. And this adduct can fit into this wide active center and can be like replicated. And something normal is put opposite it, like A, T, G, and C, and everything is fine. But sometimes it does not work. Sometimes the nucleotide is so damaged that these polymerases don't know which nucleotide it was, and so they can't put a correct nucleotide. They just put something. So it sort of looks like the saucer repair in bacteria. Just put something, so there is no replication arrest, and so we don't die. Yeah, and so
and
this
then
is
we have a mutation.
and then we have a mutation. Yeah, so it's fixed. Then we have a mutation, but at least we don't die. Maybe, hopefully, because these mutations that we're talking about are somatic mutations in the soma, in the body, so they are not inherited across generations. A mutation that you have in your body can't be transmitted to your progeny, luckily. But a mutation that you have in the germline, in the gonads, this one can be transmitted, and it's the basis of hereditary diseases. Yeah,
like many of sickness diseases, actually. Yes.
Like even sometimes, not always. Sometimes Parkinson's disease is
yes, there are genes associated with Parkinson's disease, and also
so sometimes the Parkin gene is the cause, or no? There's several causes of Parkinson's disease. I think 3 genes that I read about, they are associated with. So sometimes they are considered to be the cause of hereditary Parkinson's disease. But like, this is not a very prevalent thing. Like maybe 10% of Parkinson's disease is hereditary, but the other cases.
Yes. And all the models, all biological animal models that mimic Parkinson's disease, they're actually based on these genes. So they model only 10% of Parkinson's.
Yeah, that's a good point. What are we modeling? Yeah. What are we modeling? Yeah. So a good example of a mutation that eventually influences the central nervous system is I have a dog here, and it's a collie. And so collies, they have this MDR mutation, MDR1. So like being a collie, is connected with frequently having a mutation of the MDR1 gene, which is a component of the blood-brain barrier. You see?
Oh, yeah.
Yeah. And so whenever they have such a mutation, it is dangerous to treat them with certain anti-tick medication. And dogs that go outside a lot, they get dangerous diseases transmitted by ticks, and they can die in a few days. Dogs. So we need to protect the
They
dog.
have more permeable the blood-brain barrier, right? So actually, pathogens they can cross the barrier and infect directly neurons, for example.
Yes, but not these pathogens. I'm talking about the medication that we treat the dogs not to get as an anti-tick medications. They can permeate the blood-brain barrier and cause seizures and even death in collies. You see?
Yeah.
Components of next guard spectra, I think. Yeah, well, and so we needed our collie to be checked before it could get this medication against ticks, so that nothing could happen. And luckily, our collie was MDR1 plus, so it was not a mutant, but a certain percentage of collies have this mutation in the blood-brain barrier that can cause their bad reactions with these very frequent now medications, because ticks are prevalent.
And for cats, we also have to treat our cat, because if she's outside, then yes, of course, the risk of getting a tick is high. Wow, thank you, Nastya, for telling this, because I think all owners of collie they have to consider this fact. Yes, please check your collies, genotypes of your collies. Yes.
If you want. Maybe choose a different medication, because there are different medications, but this new one is very effective. But for collies, you need to pay attention to that.
So since we have two strings, and for example, we have a mutation in one string, but then the second one is intact, and in some cases, cells actually can choose, actually not the cells themselves, but polymerases, they can find the correct string and then work with the correct one. How does it happen? Because I think it could be a key to treat many diseases if we could manipulate with this switch mechanism. If our cells could use only intact strings.
Well, I'm not sure about this work in higher organisms, but as for bacteria, they do have this. I think this idea comes from bacteria, actually, because they have strands, they have DNA methylation that depends on the time of, that depends on how new the DNA strand is. So the old strand is methylated, but the new strand that has only been synthesized, it is not methylated yet. And so this is how a bacteria can choose the old and new, but actually, I'm not so sure about this. But the bacteria at least can choose between the self DNA that is methylated and the non-self DNA of a phage, which is a bacterial virus, a phage.
Okay.
So it has arrived into the cell, and its DNA is not methylated, and the bacteria thinks, "Aha, I know you're my attack." Attack. attack it. So the idea is not new, but how we can realize this in humans. I think this requires a lot of further study.
Yes,
Actually,
I agree. But do you agree that it could be a very big step in the future,
of
or
course.
in the medicine of the future?
If we could mark one of the strands for, but this should be done at the stage of transcription, though. So you can tell the RNA polymerase, the person that or the protein that makes copies of a gene onto the RNA template, to copy one of the two DNA strands. But it's not even on the level of a strand. It's on the level of a chromosome. So,
Epigenetics.
yes, but the initial. So then every cell has two sets of chromosomes. The one that, and every chromosome is the one that came from the father and the mother, and we can in the future hopefully ask to transcribe just one of these chromosomes, the one that is healthy, for example. But as of now, I do not know how to direct this transcription request onto one of the chromosomes. I'm not sure how we could do that.
But I'm pretty sure that since it was reported that it exists naturally, but for now we don't know how to explain it. We just know that some cells in some tissue, including the brain, that they have preferences in transcription from mother's chromosome or from father's chromosome. So maybe with time we will understand how to use it and benefit from this. But you've just mentioned viruses. I think this is a notion or maybe a topic for the next episode with you. But since viruses have the ability to actually go into DNA, right? And to how could I say it? To intersect. Yes,
to
integrate DNA. So what happens with our repair system? How do viruses just bypass it?
Repair. Okay. So I did not tell you about a group of repair mechanisms. So we talked about direct repair, base excision repair, nucleotide excision repair, but there exists another group of repair mechanisms that repair double-stranded breaks, DNA double-stranded breaks. This is another type of DNA damage, and it is also caused by certain chemotherapy agents, like for glioblastoma, it's lomustine. So lomustine is also an alkylating agent, but eventually it damages DNA so hard that it breaks into DNA double-strand breaks. And so DNA double-stranded breaks, they are repaired by several mechanisms, which are non-homologous end joining, microhomology mediated end joining, and homology repair. Homology repair. And so I believe, I'm not sure exactly about this, that viruses can harness some of these mechanisms to integrate into DNA. but also there are types of viruses that have their own integrases.
This is a type of proteins, right, that facilitates their integration into the host DNA, right?
Exactly. And so when they get inside the cell, they need to express their proteins. They need to express their proteins to make them actually from their own DNA that will allow them to integrate into the host DNA. And also there are viruses that can viruses have a variety of genomes. They can be DNA. They have DNA as their genetic material, but also RNA. RNA viruses, and some of them can have a different type of integration into DNA. But I think we shouldn't delve too deep.
But yes, viruses are smart. They're not as smart as bacteria are, but yeah.
And they're very diverse. Yeah, so it's like virology is a different branch of biology.
Let's sum up how cancer treatment works, because as I understood from what we've discussed now, is that since repair mechanisms are very weak in cancer cells, this is actually an advantage in treating cancer cells, because drugs induce DNA damage, and the repair system doesn't cope with this damage, and that's how cancer cells die, right? And that's also how not cancer cells survive, because in their cases, repair mechanisms and mechanisms that are involved in this DNA damage replication, they actually help the cell to survive, right?
So you're talking about chemotherapy for cancer.
Yes. For example.
I believe that cells that have their repair and DNA damage tolerance systems turned off are sort of terminal, are sort of cancer at some terminal stage. So usually doctors, they do not rely on the fact that repair systems are turned off. They don't. Yes. They believe that they're working. And so now there is quite a big literature, quite a literature on inhibiting DNA repair and DNA damage tolerance processes to help chemotherapy work better.
Okay.
So chemotherapy causes different types of DNA damage, like DNA alkylation, oxidation, DNA strand breaks, and so even radiation is used for example, for glioblastoma treatment. And so now different additions to chemotherapy and radiation therapy are under clinical trials to supplement these treatments with inhibition of repair and tolerance of DNA damage.
This is a good news that new methods, like additional methods that involve these mechanisms, I hope will appear soon. Because when I worked with nanoparticles, yes, we also tried to deal with glioblastoma, And I remember how it was specific for patients because the drug was already the clinical stage, and it was very heterogeneous for patients. But I believe that if we will use a more general mechanism, for example, the inhibition of the repair system, then we will have more chances to fight glioblastoma.
That is very interesting. But I think that maybe for treatments like these, for nanoparticles, maybe the future research would be to look at certain DNA markers, certain genetic markers of predisposition to such nanoparticle treatment. Or maybe
yes, this is my big dream. Please hire me. Yes.
Yeah. That would be great because there are not too many genetic markers that could tell us about the preference of this or that treatment for glioblastoma, or of this or that even for diagnosis. There are not too many genetic markers, and
Yes.
not much for prognosis, for example. It's a great field that requires further investigation.
Yes, this is already about individual medicine, so patient-based medicine. So this is where we analyze biomarkers and specific biodata from each patient, and then try to find the same patient possibly from another country or somewhere, and then we can compare, okay, this treatment worked well with this patient with exactly the same profile, then let's try the same protocol for the new patient. Yes, I believe that the future of science actually and medical research is based on this. The final one. Since we are talking about future of science and biomedical research, could you please name 3 things that would help to improve science and maybe quality of research, or maybe quality of researchers' life, for example? 3 things that you think are missing now but are very important.
That is a very large question. Could you give me an example, maybe?
So I think, if we will continue this topic about biomedical and personalized medicine, I would say that it would be great to do open data sets. This is what I saw, and this is what I use for electrophysiology as neurobiologist, and it is really helpful now because currently, a lot of things are already measured, experimentally, and we have huge data sets. And maybe during working on your dissertation, you was focused on one aspect, but other aspects, they're still in your data, and you can share your data, raw data, for other people. I think that one aspect of this improvement of science would be the creation of open data sets. What do you think?
Well, at least in the field of Drosophila omics, the data is open. So you can download anything from any paper. The genomes, the transcriptomes. So I think it's mostly with human data.
Human data. And this is what we need the most for the better treatment, for example.
Yeah,
the next thing will be from you.
Yeah, that would be wonderful. And I would love journals that publish negative results, actually.
Oh, yes!
Because we lose so much. I believe we repeat probably so much what others have done without knowing that it will not lead to to anything that would not need to be done if it had been published, but it's not. And this is one
agree,
big
like
thing.
100%, yes, that the negative result is the result too. And first of all, it will be helpful to understand that this strategy or that this hypothesis actually they didn't work upon some conditions. It would be super useful because as a researcher, you understand it, and you also faced it that you find a paper where everything is splendid and super cool, and then you try to reproduce this data in your lab, and then you see, for example, that animals die upon these conditions, and you can't continue to use the same protocols, and you check the concentrations, and you find out that actually this is a very, I don't know, severe concentrations, and I don't know how these people published this paper, right? Because they needed to show positive results, and maybe they excluded 90% of animals of the research or something like this. But this is a big problem, and this is the reason why we have very low reproducibility of results in biology and biomedicine. Yes, journals that would publish negative results. Cool. Yes.
Yeah. Also, I feel maybe this is not. No, but science will definitely benefit from a better connection with doctors, or maybe doctors that are also scientists. More of these, please, because it is such a pain that when you come. Like, I've had several episodes of dealing with different cancer cases among my the people I know, you see, and it is such a pain to talk with doctors sometimes because we don't understand each other. Although I am a biologist and I should understand everything they say, but we don't understand each other. So maybe education of people who are both biologists and doctors, medical workers.
Medical workers. Do you mean the medical staff, right?
Doctors, you see, oncologists, maybe.
Oncologists, maybe.
First of all, yeah, because I'm not sure that oncologists. Well, an oncologist who could think like a molecular biologist would be gold. That's what I think. More of these, please. We really need them.
Yes, I got it. But also, like, yes, you mean this communication.
All the mechanisms.
More open communication between doctors and medical staff and biomedical researchers. Yes, I agree with you totally, because that's really sad to understand, for example, that, I don't know, the topic that you have been investigating the last 4 years is not very helpful upon real conditions in the hospital, for example, or something like this, or actually that you already can provide useful data for doctors to improve conditions in patients already. This data exists.
Well,
yes.
Well, I understand that it is hard for doctors to use this type of data because it's not in the recommendations yet. And so, of course, I realize that it is a big way until data comes to doctors in their recommendations and they can really use it. But at least I would love doctors to approach biologists and ask them all about DNA and maybe make seminars or like your channel is a very beneficial thing for doctors to watch. Like, I would love to answer any questions from any doctor, really, like about stuff that I understand, like DNA and mutations and like the mechanism of chemotherapy action or something like that.
Yes, if you are a doctor and you have questions to Anastasya about topics that we've been discussing this hour, then please write a comment and I will give you her contact.
Okay, but thank you. But even to Vasilisa, if you have questions to Vasilisa, she can make a new video, maybe.
Yes, please share, like, follow, subscribe, and don't forget our Instagram page. This is the page where me and my cameraman were actively communicating with our audience, and this is where I ask you about your preferences, problems, so I could make new episodes and find researchers for you to explain the topic that is interesting exactly for you. So thank you, Nastya, very much for your time, for your interesting talk. And finally, imagine, I'm 30 years old. Finally, I'm starting to understand how DNA works.
I'm very happy, Vasilisa. Thank you.